Hello, so my name is Angel. Let's study Introduction to Immunology together. Alright, so let's find out what is immunology. The word immunity comes from the Latin word immunis, which means exempt. Immunology is a science that examines the structure and function of the immune system. Immunology refers to study of immune system, a complex network of interacting cells, cell products, and cell-forming tissues that protect the body from infectious agents and other foreign substances. It originates from the medicine and early studies on the causes of immunity to disease. How does it work? The immune system uses a variety of defense mechanisms to protect against infections. When a host encounters a foreign organism, the immune system responds in a way that depends on the host genetics, the type of organism, and the environment, right? So this is the immune system: macrophage, neutrophil, infected cell, antibodies, B cell, dendritic cells, and bacteria, right? They're all comprised of immune system. What happens when it doesn't work? Well, let's say your immune system stops working. What happens? So when the immune system isn't functioning properly, it can lead to disease such as allergies, cancer, or autoimmunity. Immune responses may also contribute to other disorders such as Alzheimer's, right? Cardiovascular conditions, and metabolic conditions. What are the different branches of immunology? Testicular immunology, immunodermatology, intravascular immunology, osteoimmunology, mucosal immunology, right? Who is an immunologist? Immunologists are doctors who specialize in the immune system. They can diagnose and treat a variety of immune system disorders, including allergies, asthma, immunodeficiency disorders. Immunologists may also conduct research on the immune system. So let's find out what's the history of immunology. The earliest known reference to immunity was during plague of Athens in 430 BC. Thucydides noted that people who had recovered from previous bout of disease could nurse the sick without contracting the illness a second time, right? So it talks about adaptive immunity. Observations of acquired immunity, which is adaptive immunity, were later exploited by Louis Pasteur in his development of vaccination and his proposed germ theory of disease. In 1891, Robert Koch gave proofs for which he was awarded a Nobel Prize in 1905 that microorganisms were confirmed as the cause of infectious diseases. Viruses were confirmed as human pathogens in 1901 with the discovery of the yellow fever virus by Walter Reed. Immunology made a great advance towards the end of the 19th century through rapid developments in the study of humoral immunity and cellular immunity. What are the different types of immunity? Alright, so one is an innate immunity. Organs, tissues, cells of the immune system that you're born with. Example, skin. Most of the anatomical barriers are your innate immunity. So other type of immunity is acquired immunity, or you can call it adaptive immunity, right? Immunity that develops during your lifetime, right? Active immunity and passive immunity are two types of acquired immunity, right? So what is active immunity? Active immunity develops in response to an infection or vaccination, right? Passive immunity develops after you receive the antibodies from someone or somewhere else, right? Active immunity has two different types. One is natural, one is artificial. Natural is antibodies developed in response to an infection, right? Artificial active immunity is antibodies developed in the response to a vaccination. That's artificial. Natural passive immunity, antibodies received from the mother through breast milk, right? We all know that a baby is immune to certain diseases, when the baby is nursed, because it comes from the breast milk. Because the mother already has antibodies that are received from the mother. The mother already has antibodies. Artificial immunity, antibodies received from a medicine, from a gamma globulin injection or infusion. So that's passive immunity, but antibodies that is received from a medicine, right? Artificial is antibodies developed in response to a vaccination. And passive artificial immunity is antibodies received from medicine from a gamma globule injection or infusion, right? So antibodies received from any medicine. All right, so what is innate immunity? Innate immune system or non-specific immune system is one of the two main immunity strategies, the other being the adaptive immune system in vertebrates. The innate immune system is an alternate defense strategy and is the dominant immune system response found in plants, fungi, prokaryotes, and invertebrates, right? Functions of innate immune system: recruit immune cells to infection sites by producing chemical factors, including chemical mediators called cytokines, right? Cytokines are very important. We'll study about them. Second is activate the complement cascade to identify bacteria, active cells, and promote clearance of antibodies, complexes, or dead cells. Number 3 is identify and remove foreign substance present in organs, tissues, blood, and lymph by specialized WBCs. Activate the adaptive immune system through antigen presentation. Right? So they're not only recruiting, they're activating the complement cascade, which is a complement system to identify bacteria. They're also identifying and removing foreign substances. The fourth, activate the adaptive immune through antigen production. So they're also activating the adaptive immune system through antigen presentation, right? So it also has an impact on adaptive immune system. Act as a physical barrier and chemical barrier to infectious agents through physical measures such as skin, mucus, and chemical measures such as clotting, factors, and host defense peptides, right? Anatomical barriers. What are the first innate immune system defense, right? Anatomical barriers are the first. First is skin. Your sweat include dermacidin, cathelicidin, and desquamation, flushing, organic acid, and skin flora. Right? The epithelial surface forms a physical barrier that is impermeable to most infectious agents, acting as a first line of defense against invading organisms. Right? Imagine yourself without any skin. Our organs will be just squishy. They might as well just drop. Right? So somewhere I feel that skin is a very important first line of defense immune system. Desquamation, that is shedding of the skin. Epithelium also helps remove bacteria and other infectious agents that have adhered to the epithelial surface. That's true, right? That's why dermatologists or doctors in general would ask you to make sure you're using some sort of scrub when you're taking a shower. It's very important to shed, your skin. Lack of blood vessels, the inability of the epidermis to retain moisture, and the presence of sebaceous glands in the dermis produces an environment unsuitable for the survival of the microbes, right? So environment is everything for the microbial survival. So skin, because there's inability of the epidermis to retain moisture, where there is moisture, chances are you will have certain issues. I have seen witness some skins, where they have so much of moisture that it actually ends up creating a lot of issues. It can be dandruff, it can be yeast, it can be fungal, So your skin is the first line of defense, and because the skin is not able to retain moisture, it can actually create an environment that's not suitable for microbes, right? GI tract in the gastrointestinal and respiratory tract, movement due to peristalsis or cilia, respectively, helps remove infectious agents. Also, mucus traps infectious agents. Gut flora can prevent colonization of pathogenic bacteria by secreting toxic substances or by competing with pathogenic bacteria for nutrients or cell surface attachment sites, right? So GI tract also has a very important, is also acting as a very important anatomical barrier because it removes infectious agents. Also mucus traps infectious agents. Gut flora can prevent colonization of pathogenic bacteria. Gut flora, the good bacteria, protect us from the bad bacteria, right? By secreting toxic substances or by competing with pathogenic bacteria for nutrients or cell surface attachment sites. That's why many a gastroenterologist would be suggest you to eat something that has lactobacilli, right? Lactobacilli is a good gut bacteria. It not only helps in digestion, but also helps us excrete the waste that we do not need, which includes some microbial waste as well. Anatomical barriers. Ice is one of the anatomical barriers. Flush in action of tears and saliva helps prevent infection of the eyes and the mouth. That's right, right? When we cry, sometimes we feel, why are our tears salty? But the tears are salty is because it gets rid of a lot many, unwanted microbes that we do not actually need. That's why sometimes when we have tears, we'll see some residue coming out with the tears as well, right? So that happens. Saliva helps prevent infection of the eyes and the mouth. Saliva is important because saliva not only helps us with digestion, right? Because it has enzymes, but it also helps our mouth to not get it dry, right? Imagine your mouth with no saliva. It would be crazy. Respiratory airways and lungs, mucociliary escalator, surfactant, and defenses. These are all respiratory airways and lungs, right? Cilia is actually a very protective barrier because what it does, it helps helps our respiratory airways from getting contracted by microbe by stopping it at the barrier itself. So cilia is very important. Nasopharynx, mucus, saliva, and lysozymes, they are actually very important. Imagine not having mucus in your nose. Mucus is important because it traps all the debris when you inhale, right? If you live in polluted areas, you will realize how much of pollution can cause damage to your nose and then your respiration when you inhale and or exhale, especially when you inhale. Blood-brain barrier, endothelial cells, we have passive distribution, osmosis, and active selection. P-glycoprotein mechanism by which active transportation is mediated. So blood-brain barrier is also very important, especially passive and diffusion, osmosis, and active selection. P-glycoprotein mechanism by which active transportation is mediated. If you did not have blood-brain barrier, then chances are our body will not be able to manage the extra fluids in our body, right? What if there was no barrier? Blood would be everywhere, would go like crazy. So blood-brain barrier is also very important. Inflammation. Inflammation is one of the first responses of the immune system to infection or irritation. Inflammation is stimulated by chemical factors released by injured cells. It establishes a physical barrier against spread of infection and promotes the healing of any damaged tissue following pathogen clearance. Process of acute inflammation. Major processes in acute inflammation resolution. Initiation phase of acute inflammation is characterized by the rapid infiltration of PMNS, polymorphonuclear neutrophils, followed by the infiltration of monocytes that mature into macrophages and edema formation in response to the injury, right? So lymphedema can be considered one of the acute inflammation process. PMNS provide the first line of the immune defense by migrating to sites of injury and neutralizing the invading microorganism or noxious materials by phagocytosis, right? In the resolution phase, PMNS undergo apoptosis, a programmed cell death, and are ingested by macrophages, right? Macro means large, phaging means eat. So macrophages that immigrate rapidly from the inflamed site to draining lymph nodes, right? So PMNS undergo apoptosis and are ingested by macrophages. Process of acute inflammation. So this is the acute inflammation. This is the PMNS. You understand how it's immigrating and it's going to edema, where you have LND, the lymph node drainage, the blood, and then stop PMN infiltration, apoptosis, and then it goes to lymph nodes. So that's a resolution. So if you have, let's say, an acute inflammation, these workers will go directly where you have an inflammation and then cause the lymph drain stoppage, right? So they come in and they say, can you please stop we don't want any further. So you might have a swollen node, but that acts like a protective barrier to different parts of your body, especially to your vital organs, your brain, your heart, your lungs, etc. The inflammatory response is characterized by following symptoms: redness of the skin due to locally increased blood circulation, heat, either increased local temperature such as warm feeling around a localized infection, or systemic fever. Anytime when you have an illness or disease, 9 out of 10, your body will either get very warm or it will get very cold. Swelling of affected tissues such as upper throat during common cold or joints affected by rheumatoid arthritis. Increased production of mucus, which can cause symptoms like runny nose or a productive cough. Pain, either local pain such as painful joints or a sore throat, or affecting the whole body such as body aches and possible dysfunction of involved organs tissues. White blood cells, which are also called leukocytes. Leuko means white and cytes means cells. Most leukocytes differ from other cells of the body in that they are not tightly associated with a particular organ or tissue. Thus, their function is like that of independent single-cell organisms. Most leukocytes can move freely and interact with and capture cellular debris, foreign particles, and invading microorganisms, although macrophages, mast cells, and dendritic cells are less mobile, right? So most leukocytes can move freely and interact with and capture cellular debris, foreign particles, and invading microorganisms. Yeah. So mobility, leukocytes are mobile. So white blood cells actually function independently as single-cell organisms, and they're not just connected to, any organ or tissue. There is no entity connection. They're more free to move around. Unlike many other cells, most innate immune leukocytes cannot divide or reproduce on their own, but are the products of multipotent hematopoietic stem cells present in bone marrow. Right? The innate leukocytes include natural killer cells, mast cells, eosinophils, basophils, and phagocytic cells include macrophages, neutrophils, and dendritic cells, and function within the immune system by identifying and eliminating pathogens that might cause infection. Types of WBCs, right? Type of white blood cells. There are two main types: granulocyte, which literally means with grains, agranulocyte, which means with no grains. Neutrophil, eosinophil, basophil, right, are all granulocytes. Agranulocytes are lymphocyte and monocyte, right? So this helps in phagocytosis. Neutrophil helps in phagocytosis. Eosinophil fights against parasitic infection. Basophil produces inflammatory and allergic reactions. So that's basophil. Lymphocyte produces specific immune response. Lymphocyte produces specific immune response. Monocyte fights off bacteria, viruses, and fungi. Right? So there are three types of lymphocytes: B lymphocytes, T lymphocytes, and natural killer cells. What are mast cells? Mast cells, as you can see, alright, so this is a mast cell granule sac which contains mediators. That is a mediator. Resting mast cell, and this is the activated mast cell, right? Mast cells are a type of innate immune cell that resides in connective tissue and in mucous membranes. They are intimately associated with wound healing and defense against pathogens, but are also often associated with allergies and anaphylaxis, right? When activated, mast cells rapidly release characteristic granules rich in histamine and heparin, along with various hormonal mediators and chemokines or chemotactic cytokines in the environment, right? When activated, they rapidly release granules, right? That's why, as you can see, all these granules are rapidly released, rich in histamine and heparin, along with various hormonal mediators and chemokines. Histamine dilates blood vessels, causing the characteristic signs of inflammation, and recruits neutrophils and macrophages, right? So histamine, so mast cells rapidly release the granules rich in histamine and heparin, right? And histamine dilates blood vessels, causing the characteristic signs of inflammation, and recruits neutrophils and macrophages, right? Phagocytes. What are phagocytes? These are immune cells that engulf or phagocytose pathogens or particles. That's phagocytosis. To engulf a particle or pathogen, a phagocyte extends portion of its plasma membrane, wrapping the membrane around the particle until it's enveloped. That is, a particle is now inside the cell. Once inside the cell, the invading pathogen is contained inside a phagosome, which merges with a lysosome. The lysosome contains enzymes and acids that kill and digest the particle or organism, right? So, this is the nucleus. That's a cytoplasm. Entrapment of the food particles. These are food particles, this cell membrane. These are lysosomes. So that's the food vacuole. As you can see, the food is literally inside, uh, you know, inside, uh, inside the body, the phagocyte. Formation of food vacuole within the cell. As you can see, there is a formation of food vacuole. It fuses, fusion of lysozymes with the food vacuole. As you can see, the lysozymes are surrounded by the food vacuole, right? Digestion of the food particle, then it's completely digested. So it's phagocyte, right? So if there are any pathogens, that come in contact with phagocyte, this is exactly how they just eat it, like they just fag it, they eat it. Phagocytes continue. Phagocytes patrol the body searching for pathogens, but are also able to react to a group of highly specialized molecular signals produced by other cells called cytokines. Phagocytes patrol the body searching for pathogens, but are also able to react to a group of highly specialized molecular signals produced by other cells. The phagocytic cells of the immune system include macrophages, neutrophils, and dendritic cells. Phagocytosis of the host's own cells is common as a part of regular tissue development and maintenance. When the host cells die, either by apoptosis or by cell injury due to an infection, phagocytic cells are responsible for the removal from the affected site. By helping to remove dead cells preceding growth and development of new healthy cells, phagocytosis is an important part of the healing process following the tissue injury. What are macrophages? Macrophages, from the Greek meaning large eaters, macro, are large phagocytic leukocytes which can move beyond the vascular system by migrating through the walls of capillary vessels and entering the areas between cells in the pursuit of invading pathogens. In tissues, organ-specific macrophages are differentiated from phagocytic cells present in the blood cells called monocytes. Macrophages are the most efficient phagocytes and can phagocytose substantial numbers of bacteria or other cells or microbes. Binding of bacterial molecules to receptors on the surface of macrophage triggers it to engulf and destroy the bacteria through the generation of respiratory buzz, causing the release of reactive oxygen species. Pathogens also stimulate the macrophage to produce chemokines, which summon other cells to the site of infection. Alright, so as you can see, this is a macrophage, irregular nuclear contour, granular to linear chromatin, cytoplasmic granules, cytoplasmic vacuoles. Pathogens also stimulate the macrophage to produce chemokines, so it produces chemokines, which summon other cell to site of infection. So chemokines are messenger cells. They summon, hey, can you please come here? We have an infection. We need your help. Hello, hello, come here, like that. So binding of bacterial molecules to receptors on the surface of macrophage triggers it to engulf and destroy the bacteria. All right, so causing the release of reactive oxygen species. Okay. What are neutrophils? All right. Neutrophils, along with the eosinophils, basophils, are known as granulocytes due to the presence of granules in their cytoplasm. As you can see, there's cytoplasm everywhere. 1, 2, 3, 4 granules, right? Or as polymorphonuclear cells, PMNs, due to their distinctive lobed nuclei. As you can see, they're lobed nuclei, right? They have nucleus, but it's lobed. Neutrophil granules contain a variety of toxic substances that kill or inhibit growth of bacteria and fungi. Like macrophages, neutrophils attack pathogens by activating a respiratory burst. The main products of neutrophil respiratory burst are strong oxidizing agents, including hydrogen peroxide, free oxygen radicals, and hypochlorite. Neutrophils are most abundant type of phagocyte, normally representing 50 to 60% of total circulating leukocytes, and are usually the first cells to arrive at the site of an infection. All right, so neutrophils are the most abundant type of phagocyte, right? Representing 50 to 60% of total circulating leukocytes. The bone marrow of a normal healthy adult produces more than 100 billion neutrophils per day, and more than 10 times that many per day during an acute inflammation. So if you have an inflammation, chances are your bone marrow will be producing 10 times more, so 10 into 100 billion, right? Different forms of neutrophil. Neutrophil, this is squeezing neutrophil, neutrophil crawling, neutrophil degranulating, it's letting go of granules, neutrophil band cell, neutrophil apoptosis, programmed cell death, neutrophil hypersegmented, so many lobes, right? Neutrophil phagocytosing, it's trying to eat, so it's trying to eat. It's creating a food vacuole, the microbes inside, and boom, gone. It's digested, right? All right, dendritic cells, DCS, are phagocytic cells present in tissues that are in contact with the external environment, mainly the skin, right, where they are often called Langerhans cells, and the inner mucosal lining of nose, lungs, stomach, and intestines. They are named for their resemblance to neuronal dendrites, but dendritic cells are not connected to the nervous system. Dendritic cells are very important in the process of antigen presentation and serve as a link between the innate and adaptive immune system. They are more or less connection between innate and adaptive system. All right, so this is dendritic cell, immature dendritic cell, pathogen cytokines, it gets mature, right? And this is the mature dendritic cell, right? Basophils and eosinophils. Basophils and eosinophils are cells related to neutrophils. When activated by a pathogen encounter, histamine-releasing basophils, right, are important in the defense against parasites and play a role in allergic reactions such as asthma, right? We just read that it releases histamine and heparin, right? Upon activation, eosinophils secrete a range of highly toxic proteins and free radicals that are highly effective in killing parasites, but may also damage tissue during an allergic reaction. Activation and release of toxins by eosinophils are therefore tightly regulated to prevent any inappropriate tissue destruction. So activation and release of toxins by eosinophils are therefore tightly regulated because chances are it may also inappropriately destroy a healthy tissue. So you have to regulate it. Okay? Natural killer cells. What are NKCS, right? Natural killer cells do not directly attack invading microbes, right? This is the target cell. This is the natural killer cell. And this is the activating ligand, activating receptor, inhibitory receptor, and MHC1, right? So as you can see, it cannot start attacking just like that. Rather, NK cells destroy compromised host cells such as tumor cells or virus-infected cells, recognizing such cells by the condition known as missing self. It uses a mechanism called as missing self. All right. So this term describes cells with abnormally low levels of cell surface marker called MHC1, major histocompatibility complex, a situation that can arise in viral infections or host cells. So targeted cells should have this complex, MHC1 complex. And then the NK cell is like, oh, well, you have a complex and we need to attack you. This is not going to fly, okay? All right, so they were named natural killer because of the initial notion that they do not require activating to kill cells that are missing self, right? The MHC makeup on the surface of damaged cells is altered and NK cells become activated by recognizing this. Normal body cells are not recognized and attacked by NK cells because they express intact self-MHC antigens. Those MHC antigens are recognized by killer cell immunoglobulin receptors, KIR, that slow the reactions of NK cells. The NK92 cell line does not express KIR and is developed for tumor therapy. So NK cell also are important for tumor therapy. What are viruses? Viruses are small germs, pathogens that can infect you and make you sick. They can infect humans, plants, animals, bacteria, fungi. Each one infects only specific type of health. Okay. Viral infections in humans can cause no symptoms or make you extremely ill. Types of diseases that they can cause include respiratory illnesses, diarrhea and vomiting, sexually transmitted infection, STIs, and skin conditions. Okay. A virus is a small piece of genetic information in a carrying case, a protective coating called a caspid. Viruses aren't made up of cells, so they don't have all the equipment that cells do to make more copies of themselves. Right? So viruses aren't made up of cells, so they do not have any equipment that the cell needs to make more copies of themselves. Instead, they carry instructions with them and use a host cell's equipment to make more copies of the virus. It's like someone breaking into your house to use your kitchen, right? The virus brought its own recipe, but it needs to use your dishes and measuring cups, mixer, and oven to make it. Unfortunately, they usually leave a big mess when you finally kick them out. So they're more like squatters. Viruses are also sometimes called virions. Different types of viruses: influenza, corona, Ebola, HIV, Hep B, adenovirus. I should have added rabies because rabies is also a virus here. Alright, so we're done with innate immunity. What is adaptive immunity? Adaptive immunity, also known as acquired immune system, is a defense mechanism that protects the body from pathogens. Like viruses, bacteria, fungi, and parasites, it's made up of specialized cells, organs, and processes that respond to foreign substance and microorganisms, such as after an infection or vaccination. Okay? Adaptive immunity has several key characteristics. Specificity. Adaptive immune system responds to specific pathogens and can provide long-lasting protection, unlike innate immune system. Innate immune system is not specific. Memory. Adaptive immunity can remember pathogens it has encountered before and respond faster and more effectively the next time. Okay, this is why some illnesses can only be contracted once in a lifetime, right? Like chickenpox. I had chickenpox when I was a kid, and I never had it again. Right? That's memory. That is the mechanism of memory in adaptive immune system. Your body gets adapt to it, and your chances are your immune system will remember that, hey, you already came here. Even if you come, you're not allowed. Because you're chickenpox, and we know how to handle you. Right? Okay. Slower response. Adaptive immunity is slower to respond than the innate immune system, but more accurate. So adaptive immunity is more slow and steady, but it's more accurate, right? It can take days to weeks for adaptive immunity to produce a robust response, right? So it can take days or it can take weeks. Adaptive immunity involves several types of cells, including B cells, mature in the bone marrow and develop into immunocompetent cells. T cells leave the bone marrow and enter the thymus to develop into immunocompetent cells. Helper T cells express CD4 and help activate other immune cells. Cytotoxic T cells express CD8 and remove pathogens and infected host cells. T regulatory cells express CD4 and CD25 and help distinguish between self and non-self molecules. Who are our molecules and non-self molecules, right? Because it can also destroy healthy tissues, right? So that's what they're talking about. Cytotoxic T cells are more like intracellular between two cells. There is a pathogen, CD8 would come in and remove the pathogens intracellularly. Types of adaptive immunity. Immunity can be acquired actively or passively. Immunity is acquired actively when a person exposed to foreign substance and immune system responds. Passive immunity is when antibodies are transferred from one host to another. Both actively acquired and passively acquired immunity can be obtained by natural or artificial means. Naturally acquired active immunity, when a person is naturally exposed to antigens, becomes ill, then recovers, right? Naturally acquired passive immunity involves a natural transfer of antibodies from a mother to her infant, right? The antibodies cross the woman's placenta to the fetus. Antibodies can also be transferred through breast milk with the secretions of colostrum, right? Colostrum is an ingredient in mama's milk, right? Artificial acquired active immunity is done by vaccination, introducing dead or weakened antigen to the host cell, right? So artificially acquired active immunity is done by vaccination, for example, COVID, right? Artificially acquired passive immunity, this involves the introduction of antibodies rather than antigens to human body. These antibodies are from an animal or person who is already immune to the disease. Functions of adaptive immune system. Acquired immunity is triggered in vertebrates when a pathogen evades the innate immune system and generates a threshold level of antigen and generates stranger or danger signals activating dendritic cells. Recognition of specific non-self antigens in the presence of self during the process of antigen presentation. Generation of responses that are tailored to maximally eliminate specific pathogens or pathogen-infected cells. Development of immunological memory, in which pathogens are remembered through memory B cells and memory T cells. In humans, it takes 4 to 7 days for adaptive immune system to mount a significant response. On an average, adaptive would take 4 to 7 days to mount a significant response, right? So it's not just right away, it's 4 to 7 days. Lymphocytes. T and B lymphocytes are cells of the adaptive immune system, right? So adaptive immune system has two worker, right? T and B. Okay. Human body is about 3 trillion lymphocytes, which are 20 to 40% of white blood cells. Their total mass is about same as the brain or liver. Can you believe that? Okay. The peripheral bloodstream contains only 2% of all circulating lymphocytes. The other 98% move within tissues and the lymphatic system, which includes the lymph nodes and the spleen. Okay? In humans, approximately 1 to 2% of lymphocyte pool recirculates each hour to increase the opportunity for cells to encounter the specific pathogen and antigen that they react to. B cells and T cells are derived from the same multipotent hematopoietic stem cell and look identical to one another until after they're activated. B cells play a large role in humoral immune response, whereas T cells are intimately involved in cell-mediated response, cell-mediated immune responses. Lymphocytes represent approximately 20 to 40% of WBCs, all right? So lymphocyte has a mitochondria. A nucleus is round and large. That's a cytoplasm, as you can see. It also has ribosomes and ER. That's a rough endoplasmic reticulum. What are helper T cells? Helper T cells are known as CD4 positive T lymphocytes, are a type of WBC that are essential for the body's adaptive immune response. Helper T cells help the body fight infections by activating other immune cells to destroy viruses, bacteria, and other germs. They do this by activating B cells, activating macrophages, and activating cytotoxic T cells. Activating B cell, helper T cells activate B cells to produce antibodies. Activating macrophages, helper T cells activate macrophages to destroy microbes that have been ingested. Activating cytotoxic T cells, helper T cells activate cytotoxic T cells to kill infected target cells. So helper T cells are activating cytotoxic T cells to kill infected target cells. So there is a domino effect. Can you please do this? And then the person does it, like that. Activation. Helper T cells are activated when they come into contact with a foreign or cancer antigen. On the surface of an antigen, APC, right, antigen presenting cell. So if there's a cell that has a cancer antigen on it, helper T cell would get activated when it gets connected, right? When it gets in contact with it. It comes close and you're like, you look like you have an APC. Well, it's time to get working, all right, like that. The type of APC and the chemical signal it releases determine the type of effector cell the helper T cell will become, all right? So the minute it comes into contact, depending on the type of APC, the antigen presenting cell, the helper T cell, forms itself into an effector T cell. It acts like a shapeshifter. That's what it is. Production: the thymus, a small gland in the chest, produces helper T cell. Importance: without helper T cells, the body is unable to defend itself against many microbes that are normally harmless. Helper T cells express CD4 on their surface, which recognize two MHC proteins. Okay. Helper T cell activation and action. As you can see, an antigen presenting cell has come into contact, this MHC2, and that's CD4 plus. So as you can see, there's a key and lock thing going on here, right? So helper T cell, there's clonal selection. Few of them end up becoming memory T cells, saying, hey, we'll remember who you are next time you come in, okay? Alright, and others are converted into effector cells. Interleukin secretion, I ask, what are they doing? Alright, effector cells, few of them are secreting neutrophils and macrophages, which is non-specific defense. Others are becoming cellular killer T cells, cellular immunity. And others are actually becoming B cells, which are humoral immunity, just like we just read earlier. Depending on the effector cells, the helper cells will turn, depending on the APC, the helper T cells will turn into effector cells, right? So there are different types of effector cell that it turned into. One is a neutrophil, one's a macrophage, one's a killer T cell, one's a B cell, right? As you can see, there are antibodies here. Humoral immunity. And I will be creating another lecture on humoral immunity, and we are going to study together, okay? B lymphocytes and antibody production. B cells are major cells involved in creation of antibodies that circle in blood plasma and lymph, known as humoral immunity, right? It's a B cell. It's humoral immunity. Antibodies, also known as Ig, immunoglobulin, are large Y-shaped proteins used by the immune system to identify and neutralize a foreign object. We identify you and we are going to neutralize you, all right? Upon activation, B cells produce antibodies, each of which recognizing a unique antigen and neutralizing specific pathogens. Antigen-antibody binding would cause 5 different protective mechanisms. One is agglutination, reduces number of infectious units to be dealt with. Activation of complement causes inflammation and cell lysis, right? Opsonization, coating antigen with antibody enhances phagocytosis. Antibody-dependent cell-mediated cytotoxicity. Antibodies attached to target cell cause destruction by macrophages, eosinophils, and NK cell, natural killer cell. Neutralization blocks adhesion of bacteria and viruses to mucosa. So it neutralizes it. It blocks the attachment. If you're not getting, you're not sticking to something, then chances are you will not be that effective, right? Immunological memory, right? Which is a very strong mechanism for vaccination, right? When B cells and T cells are activated, some become memory B cells and some become memory T cells. Throughout the lifetime of an animal, this memory cells from a database of effective B and T lymphocyte. So chances are the memory is going to be stored in a database, and next time, they come into contact with the same pathogen, chances are they will like, oh, we know you, you're not as effective as you were the first time. All right, so chill, okay? Upon interaction with an antigen, the appropriate memory cells are selected and activated. In this manner, the second and subsequent exposure to an antigen produces stronger and faster immune response. So it's not that effective. You might just sneeze the second time and you'll be like, okay, well, we already know you. We don't, we don't care about you, okay? All right. So this is adaptive in the sense that body's immune system prepares itself for future challenges, but is maladaptive, of course, if the receptors are autoimmune. Immunological memory can be in the form of either passive short-term memory or active long-term memory. All right, so passive memory. Passive memory is usually short term, lasting between a few days and several months. Newborn infants have had no prior exposure to microbes and are particularly vulnerable to infection. Several layers of passive protections are provided by the mother. In utero, maternal IgG is transported directly across the placenta that at birth human babies have high levels of antibodies with the same range of antigen specificities as their mother. That includes in situ, situ, and also in vitro, right? Breast milk contains antibodies, mainly IgA, that are transferred to the gut of the infant, right? GI is also a very strong anatomical barrier. We just had a chat before. Protecting against bacterial infections until the newborn can synthesize its own antibodies, uh, you know, until the baby's ready, right? This is passive immunity because the fetus does not actually make any memory cells or antibodies. It does not make its own. It only borrows them from the mother, right? Uh, Short-term passive immunity can also be transferred artificially from one individual to another via antibody-rich serum, right? Active memory. In general, active immunity is long-term and can be acquired by infection, followed by B cell and T cell activation, or artificially acquired by vaccine in a process called as immunization, right? So it can also be artificially acquired through vaccination. For example, COVID, right? Or Hep B, or a lot many kids get measles, mumps, and shingles, etc. Immunization. The principle behind immunization is to introduce an antigen derived from a disease-causing organism that stimulates the immune system to develop protective immunity against that organism, but that does not itself cause pathogenic effects of that organism. An antigen is defined as any substance that binds to a specific antibody and elicits an adaptive immune response, right? So an antigen, short for antibody generator, is defined as any substance that binds to a specific antibody and elicits an adaptive immune response. What are vaccinations? Vaccination is the administration of a vaccine to help the immune system develop immunity from a disease, right? Acquired active immunity from a disease. Vaccines contain a microorganism or a virus in a weakened, live, or killed state, or proteins or toxins from the organism in stimulating the body's adaptive immunity. They help prevent the sickness from an infectious disease. When a sufficiently large percentage of population has been vaccinated, herd immunity results. Herd immunity protects those who may be immunocompromised and cannot get a vaccine because even a weakened version would harm them, right? So herd immunity protects those who may be immunocompromised, right? And cannot get a vaccine because even a weakened version would harm them, all right? So that's why many people during COVID decided not to get vaccinated because of herd immunity. They were all protected because there were others that were not immunocompromised and they were fully immune, to COVID, right? So even the ones that didn't were protected because others were basically. Effectiveness of vaccination has been widely studied and verified. Vaccination is the most effective method of preventing infectious diseases. Vaccines are a way of artificially activating the immune system to protect against infectious disease. The activation occurs through priming the immune system with an immunogen, right? Uh, Stimulating immune response with an infectious agent known as immunization. Vaccination includes various ways of administering immunogens, like we just interacted, we just chat, that, it can be in weakened state or a killed state, something that's completely dead, like that. Most vaccines are administered before a patient has contracted a disease to help increase future protection, right? For example, a veterinarian is always asked to get a rabies vaccination in case if he or she gets bitten by a dog, a raccoon, a cat, or whichever animal that may be carrying rabies virus, right? What are immunodeficiency diseases? All right, so immunodeficiency, also known as immunocompromisation, is a state in which the immune system's ability to fight infectious disease and cancer is compromised or entirely absent. Most cases are acquired secondary due to extrinsic factors that affect the patient's immune system. Examples of these extrinsic factors include HIV infection and environmental factors such as nutrition. Immunocompromisation may also be due to genetic diseases, flaws such as SCID. In clinical settings, immunosuppression by some drugs, such as steroids, can either be an adverse effect or the intended purpose of the treatment. An example of such use is organ transplant surgery as an anti-rejection measure and in patients with an overactive immune system as in autoimmune diseases, right? So an underactive immune system is not good. An overactive immune system, it has to be balanced. Some people are born with intrinsic defects in their immune system or primary immunodeficiency. A person who has an immunodeficiency of any kind is said to be immunocompromised, right? An immunocompromised individual may particularly be vulnerable to opportunistic infections in addition to normal infections that could affect anybody, right? It also decreases cancer immunosurveillance, in which the immune system scans the body cells and kills the neoplastic ones. So even the surveillance of cancer triggering cells is compromised. They are also more susceptible to infectious disease owing to the reduced protection afforded by vaccines. T cell deficiency often causes secondary disorders such as acquired immunodeficiency syndrome, which is AIDS. Primary or secondary immunodeficiency. The distinction between primary versus secondary immunodeficiency is based on respectively whether the cause originates in the immune system itself or is in turn due to insufficiency of supporting component of it or an external decreasing factor of it. A few rare diseases feature a heightened susceptibility to infections from a childhood onward. Primary immunodeficiency, also known as congenital immunodeficiencies. Many of these disorders are hereditary and are autosomal, recessive, or X-linked. There are over 95 recognized primary immunodeficiency syndromes. They are generally grouped by the part of the immune system that is malfunctioning, such as lymphocytes or granulocytes. The treatment of primary immunodeficiencies depends on the nature of defect and may involve antibody infusions, long-term antibiotics, and the stem cell transplantation. The characteristics of lacking and or impaired antibody functions can be related to illnesses such as X-linked agammaglobulinemia and common variable immune deficiencies. What is a secondary immune deficiency? Secondary immune deficiencies, also known as acquired immunodeficiency, can result from various immunosuppressive agents, for example, malnutrition, aging, particularly medications, chemotherapy, disease-modifying, anti-rheumatic drugs, immunosuppressive drugs after organ transplants, glucocorticoids, and environmental toxins like mercury and other heavy metals, pesticides, and petrochemicals like styrene, dichlorobenzene, xylene, and ethylphenol, right? For medications, the term immunosuppression generally refers to both beneficial and potential adverse effects of decreasing the function of the immune system, while the term immunodeficiency generally refers solely to the adverse effect of increasing risk for infection. What is hypersensitivity? Right? A hypersensitivity reaction is an extreme or unnecessary immune response that the body has to an antigen. There are 4 different types of hypersensitivity reactions. Some evidence suggests a potential 5th type, but this may be a subset of type 2 hypersensitivity reactions. Each type of hypersensitivity reaction is an extreme immune response to an antigen. Each type of reaction differs based on the type of antigen the body identifies, what type of immune response the body generates, and how quickly the body produces the response. Some people may refer to hypersensitivity reactions as allergies, as these are a form of hypersensitivity. Although people use these terms interchangeably, an allergic reaction typically refers to the signs and symptoms a person may experience, while a hypersensitivity reaction describes the immunological process that occurs in the body. Hypersensitivity reaction type 1, IgE mediated. Type 2 is mediated by antibodies that activate the cellular cytotoxicity. Type 3 mediated by immune complexes. Type 4 delayed T cell mediated. All right, not dependent on antibodies. The type 4 is not dependent on antibodies, right? It takes 48 to 72 hours for hypersensitivity reaction. All right, so IgE mediated hypersensitivity, this is degranulation, right? Allergen specific IgE, Fc receptor for IgE type 1. IgE mediated hypersensitivity. AG induces cross-linking of IgE bound to mast cells and basophils with release of vasoactive mediators. Typical manifestation includes systemic anaphylaxis and localized anaphylaxis such as hay fever, asthma, hives, food allergies, and eczema. I have allergies and hives to chemicals. There's a very good chance, type 1 hypersensitivity is the one that's acting and potentially mediated by IgE. Type 2, IgG or IgM, mediated cytotoxic hypersensitivity, AB directed against cell surface antigens, antibodies directed against cell surface, antigens mediate cell destruction via complement activation or ADCC, which is complement system, right? Typical manifestation include blood transfusion reactions, erythroblastosis, fetalis, and autoimmune hemolytic anemia. Immune complex 3B, C3b complement activation, neutrophil, right? We saw earlier different shapes of neutrophils, and neutrophils can also cross barriers of capillaries. We did have a chapter for right type, so that is type 3 hypersensitivity. Immune complex mediated hypersensitivity, right? AG, antigen antibody complex deposit in various tissues induce complement activation and ensuing inflammatory response mediated by massive infiltration of neutrophils. Typical manifestation include localized arthritis reaction and generalized reactions such as serum sickness, necrotizing vasculitis, glomerulonephritis, rheumatoid arthritis, systemic lupus, erythematosus. All right, so serum sickness, necrotizing vasculitis, right? So these are all type 3 hypersensitivity. All right, so type 4, cell-mediated hypersensitivity. Sensitized Th1 cells shown above release cytokines that activate macrophages or TC cells that mediate direct cellular damage, right? Th2 cells and CATLs mediate cellular response. Typical manifestations include contact dermatitis, tubercular lesions, and graft rejection, right? So at times tubercular lesions occur. That also includes superficial tubercle dispersion, right? Which is also a type 4 cell-mediated hypersensitivity. So antigen, as you can see, there's a key and lock. Sensitized Th1, the cytokines are released, and the macrophage is activated, right? So at times, if you're a podiatrist, you will come across a lot many patients that may have athlete's foot infection. They may have really dry skin, which can be considered as dermatitis, or other illnesses that may dry up the skin. But that is only a way of of your own immune system trying to protect the stratum corneum, which acts as an anatomical barrier to the inside vasculature and or tissues and to your body basically. So it's its own way of creating an immune system barrier. Okay. Autoimmunity. In immunology, autoimmunity is a system of immune responses of an organism against its own healthy cells, tissues, and other normal body constituents. Any disease resulting from the type of immune response is termed an autoimmune disease. Prominent examples include celiac disease, diabetes mellitus, Type 1, systemic lupus erythematosus, Sjögren's syndrome, eosinophilic granulomatosis with polyangiitis, Hashimoto's thyroiditis, Graves' disease, idiopathic thrombocytopenic purpura, Addison's disease, rheumatoid arthritis, ankylosing spondylitis, polymyositis, dermatomyositis, dermatomyositis, and multiple sclerosis, MS. Autoimmune diseases are very often treated with steroids. Autoimmunity means the presence of antibodies or T cells that react with self-protein and is present in all individuals, even in a normal, healthy state. It causes autoimmune disease if self-reactivity can lead to tissue damage. So if your own cells are actually affecting your own healthy tissues, that's what causes autoimmunity disease. It just means that you have access of something. What are cytokines? Cytokines are a broad and loose category of small proteins important in cell signaling. Due to their size, cytokines cannot cross lipid bilayer of cells to enter the cytoplasm and therefore typically exert their function by interacting with specific cytokine receptors on the target cell surface. Cytokines have been shown to be involved in autocrine, paracrine, and endocrine signaling as immunomodulating agents. Cytokines are multifunctional polypeptide hormones or small proteins, non-structured and light in terms of molecular weight, that are released by several types of cells. These proteins influence or regulate a range of biological processes such as immunity and inflammation. Cytokine proteins are soluble and are vital to our immune system. Cytokine portfolio is crucial to the functions of macrophages that act as guards to the innate immune system and bring about the conversion from innate to adaptive immunity. They act as messengers and mediate complex synergies between immune and non-immune cells such as hematopoietic cells, lymphoid cells, and various pro-inflammatory and anti-inflammatory cells that stimulate the growth of inflammatory and immune responses. Categories of cytokines. Based on their properties, their secretion effect on the immune response, cytokines can be broadly divided into following categories. Pro-inflammatory cytokines. These cytokines are associated with inflammatory response, amplification, perpetuation of the inflammatory process. Growth factors. These cytokines promote cell survival and bring about structural changes in the airways. Anti-inflammatory. Anti-inflammatory cytokines control the pro-inflammatory cytokine reaction. So there are different categories of cytokines, all right, and the secretion and the effect on the immune cytokines can be broadly divided into the following. It also depends on the properties and what impact is it going to have on the immune response. So one is in pro-inflammatory cytokines. The cytokines are associated with inflammatory response, amplification, and perpetuation of inflammatory process. Growth factors. These cytokines promote cell survival and bring about structural changes in the airways. Anti-inflammatory. Anti-inflammatory cytokines control the pro-inflammatory cytokine reaction. What are the different types of cytokines? Chemokines, interferons, lymphokines, interleukins, tumor necrosis factor, TNF cytokines. They're all different types of cytokines. Chemokines play a vital role in the migration of leukocyte cells and are related to chemotactic effects for inflammatory cells. Interferons aid the body's defense against cancer and viral diseases. Lymphokines are produced by lymphocytes. These cytokines attract immune cells such as macrophages. These cytokines are secreted by T cells that grow from stem cells in the bone marrow and control the immune response. Interleukins are a group of cytokines that act as chemical signals between white blood cell and are produced by a single leukocyte. They can act on other leukocytes and mediating interaction among cells. Specific interleukins can have a significant impact on cell-to-cell communication. All right. Cytokines are also called as messenger cells. I'm just saying. Interleukins are a group of cytokines that act as chemical signals between white blood cells and are produced by single leukocyte. They can act on other leukocytes, mediating interactions among cells. Right. Specific interleukins, in between WBCs, can have a significant impact on cell-to-cell communication. Tumor necrosis factor, or TNF. TNF cytokines are released in case an infection is detected by macrophages, specific white blood cells, to signal other cells of the immune system to trigger an inflammatory response. Functions of cytokines: modulate the balance between humoral and cell-based immune responses, induce inflammatory response, regulate hematopoiesis, differentiate and induce wound healing, and regulate certain cells population maturation, growth, and responsiveness. Cytokines frequently trigger the production of new cytokines, causing a cascading of events in which the latter cytokines affect the activity of the previous cytokines that release them. How are cytokines produced? So macrophages, B lymphocytes, T lymphocytes, mast cell, endothelial cells, polymorpholeukocytes, PMNS, right? Epithelial cells, connective tissue, adipocytes are all, types of cytokines. So here it is. These can all produce cytokines, right? Cytokine gene, cytokine producing cell. Any of these cells can produce cytokines. All you need is a stimulation, right? Gene activation and target cells, and then effects, right? So it creates like a channel between one cell to a target cell, and cytokines are messengers. Cytokines are actually going to the other cell and saying, hey, you know what, you're doing it wrong, we need to take care of you. So cytokines are like messengers. Cytokine production is not continuous, but rather occurs in response to specific stimuli like infection, tissue damage, or immune cell activation. Once stimulated, the cell undergoes intracellular signaling pathways involving transcription factors like NF-κB, which activates the genes responsible for cytokine production. So cytokine production does not just keep happening. There has to be a stimulation required for it, involving the transcription factors like NF-κB, which activates the gene responsible for cytokine production, right? Transcription activated genes are transcribed into mRNA, which is then translated into cytokine protein. DNA makes RNA makes protein, remember, right? So mRNA, which is then translated into cytokine protein. Secretion. The synthesized cytokine protein is packaged into vesicles and secreted from the cell to interact with other cells, expressing the appropriate receptor. Cell types involve T helper cells, primarily responsible for producing many cytokines, especially adaptive immune responses, right? Macrophages produce cytokines in response to pathogens and tissue damage, playing a key role in innate immunity. Dendritic cells present antigens to T cells and produce cytokines to stimulate immune responses. Natural killer cells release cytokines to directly kill infected cells. Importance of cytokines. Immune system regulation. Cytokines can stimulate or slow down the immune system, and they help promote, sustain, and terminate the body's immune response. Cell growth and survival. Cytokines control the growth, differentiation, and survival of cells. Anti-cancer activity. Cytokines can help make abnormal cells die and normal cells live together. Wound repair. Cytokines contained in platelets help with wound repair. Homeostasis. Cytokines help the body maintain a state of equilibrium, of homeostasis, right? So let's say if you ever participate in any kind of clinical trial of cancer, chances are there is going to be a side effect of a cytokine inflammation, which can either increase your body temperature or decrease your body's temperature, right? Like we said, when you kill cancer cells, and chances when you kill cells that may have cancer, you may also kill cytokines, right? Which plays a very important role in inflammation. All right, so it can stimulate or slow down the immune system, and they help promote, sustain, terminate the body's immune response, right? And homeostasis may be impacted, right? Cytokines help body maintain a state of equilibrium or homeostasis. All right. So thank you for watching. You can come back. We'll be making another lecture on humoral immunity. And then we'll go back and talk in details about different immunology topics. All right? So please subscribe, like, comment, and share. Bye.